Okay, so today we are focusing on the light independent reactions, and if it's independent, that means it does not depend on the light. So these are also known as our dark reactions. So the purpose of the dark reactions is to use energy from the energy-carrying molecules that we just made in the light-dependent reactions to make sugar, specifically glucose. And this is occurring in the syrup, so it's in the stroma of the chloroplast. So we're taking the stuff we made in the thylakoid, we're taking it out into the stroma to do the next set of reactions. So the actual process that's happening in the dark reactions we call the Calvin cycle. So the Calvin cycle itself is a series of enzymes-assisted chemical reactions powered by ATP and NADPH, so our energy-carrying molecules, that are producing 3 carbon sugars from our CO2 and our hydrogen from our H2O. So we're using these energy-carrying molecules to produce specific sugars. So if you remember, we are going to combine our CO2 with one of the H's from H2O. So in the Calvin cycle, this is going to happen twice, and then these 3 C sugars are going to combine to make one glucose molecule. So what we mean by 3C is it has 3 carbons, and we're doing it twice to make this 6 carbon structure. So we will talk more about what these 3 carbons are, but glucose has C6, so it is a 6 carbon structure. Okay, so if we look at the details or the steps of what's happening in the Calvin cycle, we're going to try to split it up into some 4 brief steps. So first we're going to have what we call the grab phase of the Calvin cycle, and we're calling it grab because it's grabbing onto stuff. So in the grab cycle, our carbon dioxide is going to diffuse into our stroma, so it's going to go out and mix into the syrup portion. Our enzymes are going to attach to CO2 to a 5 carbon structure of RUBP. We like to shorten this to RUBP because the actual name is 5 carbon ribulose biphosphate. So RUBP works much better to say. So we're going to take that 5 carbon, add the CO2 to it, and make an unstable 6 carbon molecule. So because it's unstable, we are going to need to do something to change this to make it into a stable form. So that brings us to step 2, which we call the split step. And in the split step, energy from our energy-carrying molecules of ATP and NADPH are going to add with an enzyme to break down that unstable 6 carbon sugar and turn it into 2 3 carbon sugar molecules known as PGA. And again, we are abbreviating PGA, and you don't actually have to know what the real name of it is because it is phosphoglycerate, and PGA is just much easier to say and remember. The third phase of the Calvin cycle we call the leave step. We call leave because our 3 carbon molecule of PGA is going to be converted into a different 3 carbon molecule known as G3P. And again, abbreviating because it is actually glyceraldehyde 3-phosphate. So just knowing it as 3GP is fine by me. One of these G3P molecules is going to leave the cycle and turn into glucose, and then with the second one is going to move on to our next step, and that next step we call the switch. So we're going to switch it into something else. So the remaining G3P is going to convert back into our original 5 carbon sugar of RUBP by using a phosphate from our ATP, and this is going to start the cycle over again. So I know there's a lot going on and it can be very confusing, but essentially you just need to know the 4 steps and just generally what is happening in them. You don't have to know the actual names for the things. Their abbreviations will work just fine. So I'm going to show you a picture of what this actually looks like in its molecular form, and it's going to be a very complex process that I don't expect you to understand or memorize or anything, but I just want you to see it so you can fully appreciate the process that is happening. So we have them numbered, so we can start at our step 1 here and then follow along, and it has the number of carbons all listed out for you and the actual name of each of the molecules it is that we are making. So again, if we go back to this picture, looking at our reactions, this is the same picture we looked at yesterday in class. So we have light coming in from our light reactions, as well as carbon dioxide coming in through our stromas, water coming in from the roots, and then we are in the first step, storing those energy-carrying molecules, kicking out our oxygen, and now we're taking those things with us. We're going to make sugar. So if we zoom in, we have our light coming into our thylakoid as well as water coming in from the roots. We're kicking out our oxygen. We're going to take those energy-carrying molecules with us over into the Calvin cycle. In the Calvin cycle, we're going to take in our carbon dioxide, kick out the sugar because that's what we're making. In your assignment for today, you are going to be filling in a diagram very similar to this one. So you might want to come back to this, and that will help you determine where to put these things. And before we move on completely, just as a friendly reminder, photosynthesis isn't the only option. We do also have chemosynthesis, and this is a process of an organism making its own food using chemicals, hence chemo, instead of sunlight, like photosynthesis. So this is the process that producers who can't do photosynthesis do to get energy. So this would be a lot of your bacteria are going to do chemosynthesis rather than photosynthesis.